This is a demonstration of how to use the Rapid Response 3-in-1 Spiked Drink Drug Test Kit. Note: humidity and temperature can affect results. Do not use the test after the expiration date indicated on the package. Do not use the test if the pouch is damaged. How to use the Rapid Response 3-in-1 Spiked Drink Drug Test Kit. Your kit will contain a product insert, pouches, and a quick reference guide. Each individually sealed pouch will contain a test cassette, sample dilution buffer tube, and a dropper. You will also require a timer. Read the instructions carefully before starting. A digital copy of the instructions is available at www.btnx.com/3-in-1 cassette. Remove all the test components from the sealed foil pouch. Perform the test within 1 hour. Do not use the test if the reaction pad of the GHB test has a purple color before applying beverage specimen. Peel open the aluminum foil on the buffer tube. Use the dropper to draw up some of the beverage you wish to test. Transfer 2 drops of beverage to the buffer tube. Close the lid of the buffer tube. Shake the buffer tube 5 to 8 times. Place the test cassette on a clean and level surface. Hold the tube vertically and transfer 3 drops of diluted beverage to the specimen well S of the test cassette. Avoid trapping air bubbles in the specimen well S. Set your timer to 5 minutes. Read the results at 5 minutes. Do not interpret the results after 10 minutes. Results interpretation are printed in the product insert, on the quick reference guide, and on the back of the pouch. Reading the results. Positive results for benzo and ketamine. A colored line appears in the control region, C, and no line appears in the test region. The positive result means that the drug concentration in the sample is greater than the designated cutoff for a specific drug. Positive results for GHB. If the reaction pad changes to a purple color, the test is positive. This result indicates that GHB was detected in the beverage. Note: if only the outer edges of the color pad change color, and the remainder of the pad is colorless, repeat the test with a new cassette. Negative results for benzo and ketamine. 2 lines appear. A colored line appears in the control region, C, and a colored line appears in the test region. This negative result means that the concentrations in the sample are below the designated cutoff levels for a particular drug tested. Note: the shade of the colored lines in the test region may vary. The result should be considered negative whenever there is even a faint line. Negative results for GHB. No color change appears. Invalid results for benzo and ketamine. No line appears in the control region, C. Insufficient specimen volume or incorrect procedural techniques are the most likely reasons for control line failure. Read the directions again and repeat the test with a new test cassette. If the result is still invalid, contact your manufacturer. Invalid results for GHB. If the color pad has a purple color before applying beverage sample, do not use the test. After completing the test, dispose of all used test materials in the trash.